this is going to be some heavy science. But I'm going to try and explain it to you the best I can so that you will be able to understand what this topic is all about. As I started out yesterday, understanding basic bee biology is the foundation of colony management. You have to understand the biological forces going on within the colony that impact the development of that colony. Yesterday, both Michael Smith and I talked to you about the idea that larger colonies are more productive colonies. There's a larger proportion of the population available to go to the field, and they begin building drone comb at an earlier stage in the population, and they build more drone comb than a small colony in comparison. So this idea of developing strong colonies is what this is all about, and what impact the glandular system has on the development of strong colonies. Your success is going to be dependent upon developing productive colonies. Colony development is regulated by many different factors. I'm only going to mention 3 here. Queen quality, which Roger and I are going to talk about tomorrow in our lectures. You providing adequate brood area so that the queen can be laying at her maximum rate at least 6 weeks before the beginning of your major honey flow. And colony development is also related to the nutritional state of that particular colony. And from a management standpoint, that's one aspect in which you can have an impact. If they're lacking in pollen, if they're lacking in honey, you can feed them, and hopefully then they will develop faster and stronger and be more productive for you. And we'll talk more about that as we go. Colony development is tied directly to the progression of flowering plants, indirectly the nutrition of the colony. And here we see 2 American species that are excellent food sources: crimson clover on your left and tulip poplar on the right. But the idea here is plant development, plant flowering, nectar secretion, pollen production are affected by the same factors that affect colony buildup and colony development. When you see differences in colonies, it may be due to differences of location and the floral sources that are present in that particular area. Timing is also an important consideration. When do you have to have your colonies in peak condition to take advantage of the major nectar flows or honey flows of your particular area? If your primary honey source, say, is available in July, but you don't get your colonies in peak condition until August, you've lost out. And so you've got to know what are your major floral sources, your major honey flow sources, and then develop your colonies so that they will be in peak condition, ready to take advantage of the local flows as the environment allows. So timing is also a part of your management production. So basically what we want to do today is to explain how differences in colony development occur. For instance, just as a scenario, 2 beekeepers get nukes from the same source at the same time, but 2.5 months later, they are vastly different in strength and production. I'm sure within your own apiary, you have similar sized colonies, and some are very productive, and others you don't get a drop of honey from a surplus. And so there are differences. And so today we're going to try to explain what causes those differences in colony development and productivity. Colony reproduction, development, division of labor, and productivity are under the control of the honeybee glandular system. Honeybees have two types of glands. They have what we call endocrine glands, and they have exocrine glands. Endo means inner, exo means outer. Endocrine glands produce hormones. They're released into the blood of that particular individual, and the hormones only affect that individual. So this is an important point for you to remember. Endocrine, they produce hormones, and they only affect the individual that's producing those hormones. Hormones regulate growth, regulate development, and molting, which we will describe in a few minutes. On the other hand, exocrine glands, as we defined for you yesterday in my first presentation, they secrete substances to the exterior of the organism. So that's where we get exocrine or outer, okay? Secrete them to the outer part of the individual's body. Here are some examples of some of the exocrine glands. We have the venom sac. To be functional, it has to be taken to the exterior of the organism through the stinger, okay? We have the Nasonov gland, which produces Nasonov pheromone, which is Ng in this particular diagram. Here we have salivary glands, one pair located within the head, the other pair located within the thorax. Here we have the hypopharyngeal glands, or brood food glands as they're sometimes referred to as. We have the mandibular glands. We have the tarsal glands, as we indicated, that produce footprint pheromone in my presentation yesterday. And here we have the wax glands, where wax is secreted to the exterior surface, and you'll see the tiny flakes forming on the ventral surface of the abdomen. So these are all examples of exocrine glands. What are some of the products of these exocrine glands? We have beeswax, we have venom, we have pheromones, and as we indicated to you yesterday, over 50 different chemical substances have been isolated from the brood, the workers, and the queens. We also have the production of enzymes, which are involved in the processing or ripening of honey. And we also have the production of royal jelly, worker jelly, and drone jelly. This proteinaceous jellies that are secreted by both the hypopharyngeal glands and the mandibular glands are used by nurse bees to care for the young larvae within the hive. We often just refer to it as royal jelly, but biochemically, royal jelly is different than worker jelly and is different than drone jelly. So biochemically, they are slightly different. These products that we've listed here that are produced by the exocrine glands have four basic functions in the biology of the colony: wax production, communication, and of course the key component there would be pheromones, defense, venom, and food processing or the ripening of nectar into honey. There are both pheromone producing and non-pheromone producing exocrine glands as we've described them to you. Now, let's first of all look at the hormone side, the endocrine side, and what effect it has on colony development. There are three major endocrine glands that produce pheromones in the honeybee. In the larvae, which what's the reason for the L, It's found only in the larvae. We have what is called the prothoracic gland. But then we have two glands that are found in both larvae and adults, the corpora allata and the corpora cardiaca. All right, so these are endocrine glands producing hormones, and as you recall, we stated they only affect the individual that is secreting them. The prothoracic glands, as I indicated, is not present in adult bees, and it's a small leaf-like structure found between the first and second thoracic segments of a young larva. The prothoracic gland produces ecdysone that controls molting in the larva and pupa. Let's first of all try to understand what molting is all about. Honeybees have an exoskeleton, an outer skin, so to speak, and this does not grow. So as the larvae internally grows, they periodically have to shed that skin. And it's under the control of the ecdysone that controls molting. And so when they shed a skin, then they can get larger. They secrete a new outer skin, so to speak. They get larger, and this is how they grow over time as they are developing. And this is all during the developmental phase of the insect. The corpora allata are two globular organs found on the sides of the esophagus behind the brain. And then the corpora cardiacum or corpora cardiaca is a loose body of cells attached to the wall of the aorta. The aorta is the heart of the honeybee. It's in the dorsal part of the body, and it pumps blood from the posterior part of the organism to the front part of the organism, both in adults as well as in larvae. Honeybees have what we call an open circulatory system. So it's just like a tank truck, so to speak. Think of a larva or an adult as a tank truck, and we have liquid blood, or we call it hemolymph, throughout the body of the bee. The heart, or the aorta, as we indicated, pumps it from the rear portion of the organism to the front part. It opens up just behind the brain, and then slowly that blood or hemolymph works its way backwards and then is repumped forward. And we call it a heart, but it has by the name of aorta. Just to show you what we're talking about here, the corpora cardiaca is located right here. This is the heart or the aorta that we were talking about, and these are the corpora allata. The function of the cardiaca are not known, and just below this structure that you see right here below the heart, this would be the esophagus. And this is the brain of the bee. So this is where they're located, directly behind the brain. But there are also nerves connected to these that go into the brain. And so the control of the brain is going to control then the secretion of the hormones from these two structures. As we say, the corpora allata are connected to two other endocrine organs by nerves. The corpora cardiaca, neurosecretory cells of the brain. The brain regulates the functioning of the corpora allata activity via neural and neuroendocrine signals. Neural signals are electric impulses traveling through the nerves of the bee. Neuroendocrine signals are chemicals that are passing through the nerve system. Endocrine means inner, okay, so you can think of them as pseudo hormones. So together with these electrical impulses and these neuroendocrine chemicals, this controls then the release and secretion of the hormones in the developing larva as well as in the adult bee. The corpora allata produce juvenile hormone. And somebody in the audience yesterday wanted me to answer or talk about juvenile hormone. And I said we'd wait till today. That is the primary hormone that's produced in both immatures as well as adult honeybees that regulates regulates development of the individual, and in the case of adults, regulates division of labor, regulates the task in which they are going to be performing. Juvenile hormone regulates metamorphosis, reproduction, and behavioral development. So this hormone has many, many different functions in relation to colony development and production. The amount of juvenile hormone that's secreted by the corpora allata is proportional to the size of the glands. The corpora allata of the queen is much larger than those of the worker larvae. And so if we were to take hemolymph samples or blood samples from workers and from queens, we would find the titers of the hormone are much higher in the queen than they are in the workers. Juvenile hormone in concert with ecdysone, which we said is the molting hormone, controls growth and development of larvae and pupae. So yesterday we said colony development is related to queen oviposition, the number of eggs she lays. All right? But what we're saying is the development of those eggs after they hatch and as they develop into adults is under the control of juvenile hormone as well as ecdysone. Developing bees undergo 6 molts. And as we said, when an individual molts, they shed their outer skin with each molt. So that means during development, this is going to happen 6 times. There are 5 molts during the larval stage. As we indicated to you yesterday in the worker caste, the larval stage lasts for 6 days. So that means if there are 5 molts during that larval period, a little over every 24 hours, they're going to shed their skin and move to the next larger level. The last molt occurs when the adult bee emerges. As brood cells are used over and over and over again, generation after generation of brood, the diameter gets slightly smaller. Why? Because the last skin that is shed is cemented, so to speak, to the inner part of the cell. And so if you looked at a cell in cross-section that's raised many generations of brood, you would see all these layers of these shed skins from each adult that emerged from that particular cell. Also, during development, they do not get rid of their waste. But at the time of their last molt, they defecate, and then the defecation is covered up with that last skin that is shed. And so that's how they keep the brood cell sanitary as that individual develops. Whether that individual, that larva as it's developing, molts to a larger larva or proceeds to the pupal stage depends upon the balance between ecdysone and juvenile hormone. So as we say, it's under the complete control of these 2 particular hormones. Division of labor. This is referring to the worker caste. Who's doing what when? Okay? As a bee ages, they move from one task to another. Whether they're from cleaning cells or whether they're accepting nectar as it's being brought into the hive, or they're guarding the front entrance, or they're nurse bees. ultimately then they will become foragers. So juvenile hormone is involved in deciding the tasks that the bee is going to be performing at any given time, or develops the division of labor. Each member of the colony has a definite task to perform, but it takes the combined efforts of the entire colony to survive and reproduce. And as we tried to show you, this is under the control of these hormones that we've talked about. This division of labor is controlled by two different factors: the age of the worker and the colony needs. Now typically, I'm just going to use two examples. As the bee ages, as the worker ages, first of all, she's a nurse bee. That means that the mandibular glands and the hypopharyngeal glands, or brood food glands as we sometimes call them, are fully developed. All right, after she completes her nursing activities, they degenerate. They don't become totally vestigial, but they do generate, lose their function. Then possibly the next gland that's going to kick in is going to be the wax glands. And so they're secreting wax and building comb. All right, now, let's say something drastic happens to your particular colony. You still have an excellent queen laying at full rate, so to speak, and whatever happened affected the size of the nurse bee population. And there's no longer adequate numbers of nurse bees to care for the larvae that are hatching from the eggs that the queen is laying. So old nurse bees, who may even be foragers by this time in their life, may go back and regenerate the function of their hypopharyngeal glands and their mandibular glands because there's a need. So normally we have a natural progression based on the aging of the bee, but if there's a need for a particular gland function in that colony, they can revert back. Old nurse bees, in comparison to young nurse bees, are not as good, do not produce as much food, but they can produce, and they can reactivate their glands. So juvenile hormone influences the pace of the behavioral development of the worker caste. This is involved in the transition from a bee being a house bee to becoming a field bee or a forager, and this is under the control of juvenile hormone. Another chemical, this is a lipoprotein. What do we mean by lipoprotein? Lipo, or lipids, are fats. And so they would be protein molecules with fat molecules attached to them. Vitellogenin production is regulated by juvenile hormone. This is a very, very important chemical associated with queens. Normally when we talk about vitellogenin, we talk about it as being a female-related chemical. So it's involved in the production of eggs and oviposition. There's less vitellogenin in workers than we find in queens because they're not necessarily producing eggs. Also, possibly you've heard there's a difference physiologically between a summer bee and a winter bee. Okay? Vitellogenin is produced within the fat body of the bee. And as summer progresses and fall approaches, then the worker caste stores vitellogenin, and that's why we call them winter bees, because they can even raise a couple of generations of brood just from the fat body that's stored within their bodies if it is absolutely necessary. But vitellogenin is regulated by juvenile hormone. As I said, this is a female type hormone, but recently they have found vitellogenin in drones. We don't know why yet. We haven't figured out why yet. But it is all, even though it's a female hormone, it is also found in the male caste. Vitellogenin is an egg yolk protein that is synthesized in the fat bodies and transported into the queen's eggs during maturation, or during when the eggs are developing and maturing. So this is the primary function of it. Vitellogenin has multiple roles, as we explained. It can be a reserve food supply found in workers. It's also a component involved in the immune system of the honeybee. It's an antioxidant to prolong lifespan, and it regulates foraging behavior. So vitellogenin has many, many functions or roles in the life or in the physiology of the honeybee. As we said, it's a storage protein that builds up during reduced brood rearing. And as I say, I've already explained the difference between summer bees and winter bees. But this is what is going on. It's a storage protein. Proteinaceous jelly, royal jelly, is a complex substance produced by two different glands. Of course, this is important in regards to nurse bees. And as we indicated, there are actually many times, I'm sure we think, based on what we read, that it's the hypopharyngeal glands or the brood food glands that are responsible for producing the royal jelly. But the mandibular gland also is involved. And this just shows you what the royal jelly looks like. You can see the tiny C-shaped larvae lying on a bed of royal jelly. It's a white, milky substance. But what we want you to realize, the source of that jelly is coming from two different glands rather than one. Hypopharyngeal glands are undeveloped when workers emerge from their cells. Within 6 to 12 days, these glands become active and develop. But in order for that to occur, nurse bees have to consume large amounts of pollen or protein. And so this is important in your considering why is one particular colony building up faster than another colony. It may have to do with the amount of protein or pollen that's stored within that colony. So protein or pollen consumption is necessary for the hypopharyngeal glands to fully develop. Pollen, it's their protein source of amino acids, which are the building blocks of proteins, minerals, vitamins, fats, and sterols. And that, in combination with nectar or honey, then gives the bees a balanced diet. Food-producing glands play a very, very important role in bee nutrition and caste differentiation. And I suspect many of you were unaware of that, but they are involved in the differentiation between the castes. The paired hypopharyngeal glands are in the front part of the head. They are looping chains of oval glandular lobes called axini, numbering over 550 on each side. So think of strands of grapes, so to speak, each grape being an axinus, all right? These lobes that we're talking about. So we got 550 lobes on this side in the head of the bee and 550 or so lobes on this particular side as well. The hypopharyngeal glands secrete the clear component of brood food, which is rich in enzymes, lipids, vitamins, and proteins. So even though royal jelly, as we look at it in a queen cell or in a worker cell, appears to be milky in coloration, it's the clear component that's coming from the hypopharyngeal glands, and it's the milky or white component that's coming from the mandibular glands, which we'll talk about next. Mandibular glands produce the white component of royal jelly. It's rich in lipids or fats, and the primary component of the mandibular gland is 10-hydroxy-2-decanoic acid. This is also another component of pheromones, okay? And I'm sure there's some relationship between the jelly and between pheromone production, etc., but we're not going to delve into that today. Even though this is not a good photograph, this is the mandible of the bee, this is the mandibular gland. In honeybees, the mandibles go like this, okay? And what we're saying is, in this area, that's where the gland is located. They secrete the milky portion or the white portion of royal jelly, and it moves down through the mandible into the mouth area or the oral area of the bee. So the ratio of hypopharyngeal to mandibular gland secretion deposited varies depending on the larval age, the sex, and the caste. This is the idea where we use the terms royal jelly, worker jelly, drone jelly. There are differences. So when a nurse bee approaches the larva to feed it, and as we indicated yesterday in our lecture, 2,000 to 3,000 nurse bee visits over a 6-day period in the worker caste, the nurse bee has to determine the sex of that larva. Is it a drone or is it a female? Has to determine the age, and then feed it the appropriate type of royal jelly. You might think from what I just said that implies that they're thinking. No, they're not. A particular nurse bee is going to be feeding a particular class age class, as well as a particular caste type as she continues her nursing activities. Now she may change as she conducts her nursing activities. Initially she may just be mass provisioning the cell as the egg hatches. And then she may do individual nurse bee visits. As she ages, she will switch from mass provisioning cells to individual visits to the developing larvae. The composition of royal jelly initially fed to young larvae is different from that fed at a later stage. Royal jelly fed to the queen larva varies in composition with larval age. The first 3 days in the development of the queen, mostly the white mandibular gland component. Days 4 and 5 contains more of the clear hypopharyngeal gland component, which is extremely rich in proteins. Worker larvae are progressively provisioned, worker jelly, a 2 to 9 to 3 ratio of mandibular to hypopharyngeal gland secretions to pure pollen. So as the larva gets older, we see a change in the ratio of hypopharyngeal glands, mandibular glands, and pollen then becomes, in the latter stages of development, pollen then becomes an important component in that jelly that they are receiving. Male larvae are provisioned food of lower protein quality, but greater quantities due to the larger size and longer development time. So the differences are expressed here between worker larvae and drone larvae. Lower quality, but more of it. They're not as important to the life of the colony. Royal jelly or queen jelly is 34% sugar. Worker jelly is only 12% sugar. Drone jelly, we don't know exactly what it is. Some of the work that's been done has been inconclusive, and so we don't know for sure. The differences in sugar, as we indicated to you, jelly is involved in caste formation. And the primary difference has to do with the sugar composition of the jelly that's being fed to either the worker larvae or to queen larvae. We use a new term up here, phagostimulant. What does that mean? Well, these are chemicals associated with the jelly that's being produced. It increases the appetite, stimulates the appetite, so that a queen larva will consume a lot more food than a worker larva. Okay? So that's what we mean by the idea that the sugar, hey, we all like take in sugar, don't we? Well, what we're saying is the increase in sugar in the diet of the queen helps them to consume more and to develop faster and larger. Presence of brood is the largest factor affecting glandular activity. As workers age, hypopharyngeal glands degenerate, and we've already talked a little bit about that. If there's no young bees present to feed, foragers can redevelop their glands, and as I say, I went through that with you. All right, now, so we said hypopharyngeal glands are responsible for producing the clear component of the proteinaceous jelly that's being produced. But once the bee stops being a nurse bee and becomes a field bee, the hypopharyngeal glands now begin producing carbohydrate metabolism enzymes. These are enzymes that are involved in the processing of nectar into honey. Examples would be alpha-glucidase, amylase, glucose oxidase, and invertase. Invertase is also known as sucrase. Takes the 12 molecule carbon molecule of sucrose and breaks it down into 6 carbon molecules, either fructose or glucose, and that's part of the ripening process of honey. And it's the enzymes that are responsible for that are produced by the hypopharyngeal glands. Alpha-glucosidase is needed for the processing of nectar into honey. Amylase converts starches that are found in nectar as part of the ripening process. Now, the other important aspect of this, understanding the glandular activity of bees and what function it has, glucose plus glucose oxidase produces gluconic acid and hydrogen peroxide. It keeps dilute nectar and syrup from fermenting. So products of the glands ultimately end up producing hydrogen peroxide, which is involved in, as I say, keeping the nectar or the syrup from fermenting. As we said, invertase inverts sucrose into glucose and fructose by splitting the bond that holds them together. So we see a change from the hypopharyngeal glands of nurse bees to the hypopharyngeal glands of foragers. Likewise, the mandibular glands. We said it involved in secreting the milky component of proteinaceous jelly. But once the bee becomes a field bee, those glands now produce a compound called 2-heptanone, which is also an alarm pheromone. This particular alarm pheromone is not nearly as effective as the alarm pheromone associated with the stinger, which is isopenyl acetate. But this is 2-heptanone. So we see these glands change as the task of the bees change as well. The nutritional condition of the colony is going to be extremely important for you to consider as you try to understand bee development. The protein contents of pollens varies from anywhere to 10 to 36% protein. So what's the take-home message? Not all pollens are nutritionally alike. Bees generally collect and utilize a mixture of pollens. Many individual pollens are nutritionally inadequate, lacking in certain amino acids required by bees. What does this boil down to? As you examine your hives, you need to see a diversity of pollen coming in if it's going to be effective in allowing colony development to progress at its maximum rate. A total of 10 amino acids must be present in the diet of honeybees for maximum development. 10 amino acids. Not all pollens contain all 10. That's why it's important that you have a mixture of pollens. If I were to take dandelion pollen, everybody know what dandelions are in here? Okay. If you were to take pure dandelion pollen and try to raise bees, they would be unsuccessful in completing development because dandelion pollen lacks some of the 10 essential amino acids. Here are the actual 10. I don't expect you to remember it, but I just want to show you that there are 10 that are absolutely necessary to complete bee development. So ideally, when you go into a colony and examine it, what we want to see is a great diversity of pollen. And of course, different colored pollens here indicates that there's a great diversity. This buffet of pollen, so to speak, is hopefully going to supply the 10 essential amino acids that are required for bee development. All right, so from a management standpoint now, if you want to have a colony developing at its maximum rates, first of all, you need to provide ample room, because that's where they store their food reserves. That's where brood rearing occurs. That's where the handling of nectar occurs as it's coming in during a honey flow, and the storage of surplus honey. I think I mentioned yesterday, nectar may be anywhere from 60 to 80% water. Final product, honey, is on the order of somewhere between 16 and 18% water, or ideally 17%. Okay? As the bees are in the midst of a major flow, all that nectar is coming in, they have to temporarily store it somewhere until they get around to processing it and ripening it into honey. They have to store all that excess water, so to speak. And that's why it's so important that you provide adequate space for your bees at the time of a major honey flow in your particular area. Having adequate comb is also extremely important. Here we see down near the bottom in this area, we see new comb, light in color, almost white in color, that was just recently built. The queen, as you see here, she must have places to lay her eggs, but they must also have places, as shown here in the lower right-hand corner, where they're going to store that honey as it comes in. Again, new, freshly secreted wax up here as well. Beeswax is produced by metabolizing honey in fat cells. And we showed you where those wax glands were located in one of our early diagrams. So we said the functioning of these glands is extremely tied to pollen diversity in the colony. But having adequate supplies of honey to produce the wax so they can build adequate supplies of comb is also an important consideration in your management scheme. The wax particles are produced by the glands in the ventral part of the abdomen here. They're pure white in color as they're secreted and hardened as they're exposed to the air. And of course, then the bees form festoons of workers that pass these wax particles to the bees that are actually building it. They're chewing it, getting it ready to build comb. And of course, this is what fresh, new, white wax looks like as it's being constructed by the bees. As we indicated to you yesterday, virtually all bee activities are directly stimulated and coordinated to a large degree by pheromones. Pheromones is another product of exocrine glands. We spent some time talking about it yesterday. We're not going to take a lot of time to talk about it today because I'm just about out of time. We defined a pheromone for you. It's a chemical substance produced by the bee, secreted to the exterior surface, and it impacts or affects the behavior or physiology of other members of the same species. Okay? Doesn't affect the individual that's producing it, but affects the other members of the colony. We told you there are two different classes of pheromones. There are releasers that stimulate an immediate behavioral response. We gave you examples of the Nasinoff gland yesterday, as well as the alarm pheromone that's produced by the stinger. Primers act slowly, cause changes in the bees that are exposed to those pheromones. In a week or so, their physiology changes, their behavior may change, and it affects their development. What is our take-home message today? And I realize it was pretty scientific and it was highly complex. Hopefully you followed me most of the time and you understand partly what we're trying to say. The take-home message is that colony development and productivity are under the control of both endocrine and exocrine glands in concert with queen quality, the size of the bee population in the hive, the amount of comb that's present, the nutritional state of that colony, as well as environmental factors. So many, many factors are going to affect the development of what we want, a productive honeybee colony.
Thank you. In the case
Can you stand up, please?
In the case of sac brood virus, which prevents the larvae from completing its fifth molt, is it actually attacking the gland? Did you say exodyso gland? Is it?
Dysone?
Yes, dysone.
Yes, it does affect the gland that produces ecdysone. It does affect it, but there's also an accumulation of liquid or water between the skins of the bee that also impacts or prevents them from shedding the skin or molting. Yes.
The winter bees live for 5 months or so, and it's a kind of very difficult time in March when the colonies kick off again. What's happening about all those glands that surely will have atrophied over the course of the winter? How come they are able suddenly to be able to produce brood? Or is it just the few bees that have been raised over the winter period that are having the burden of kick-starting the colony in the spring?
It's really a combination of both. The new bees that are produced once the queen begins laying a few eggs during the course of the winter, those new bees will have an active role in colony development and in development of brood. Also, another characteristic of a winter bee is their hypopharyngeal glands and their mandibular glands do not degenerate. They remain, they're not active in secreting, but they remain fully developed, and once the nutritional state of the colony allows, then they will kick in and be part of this jump start of the colony in the spring.
If you have a colony, say in October, that completely the queen goes off lay and there is no brood as such, does that mean tickets in March basically?
No, it does not. What it means is that the winter bees, the bees have stored a lot of fat body. I could be a great winter bee. They've stored a lot of fat body. As I say, their hypopharyngeal glands and their mandibular glands do not degenerate. They remain active, even though during the broodless period they're not going to be secreting proteinaceous jelly, but they're ready to go once the queen begins laying and there's a need for them to produce the royal jelly.
I'd like to ask, is anyone looking at pheromone distribution inside the hive over time?
Yes, there are people looking at it, like we talked about lickers and attenuators yesterday. That was all work that was done probably 20 years ago. But people are continuing to look at it and trying to get a feel for what is really going on and what does pheromone distribution within the colony really mean in regards to the biology of the colony and the division of labor.
Who's most active in that area?
who's most active in that area. Gene Robinson at the University of Illinois has been doing a lot of work in that area. There are others. I'm sure there are several European scientists that are working on it as well.
You mentioned about dandelion pollen not having the sort of 10 enzymes. Are there any particular pollens which you think are superfoods, or you've identified as superfoods, that would be particularly good for them as they start in their sort of start their season?
I don't know of anybody that has identified what we would call super pollens that contain all 10. The key here is that there's a mixture or diversity of pollen within the hive so that all 10 will be adequately supplied.
Any particular pollens then? Okay, they might not produce all 10.
I can't tell you right off the top of my head.
Okay.
Again, there's been very limited pollen analysis in regards to their amino acid content, which should be done if we're going to totally understand the nutrition of the colony.
Just interested in the mechanism by which the glands change function from producing one thing to another. What's happening inside the gland to do that?
There has to be a hormone involved in causing them to become inactive, to degenerate, to even become almost vestigial, okay? But then the same glandular tissue, again, this is undoubtedly under hormone control, switches, and they begin producing different chemicals, like the 2-heptanone of the mandibular glands and the enzymes associated with honey processing in the hypopharyngeal glands. It's all under hormonal control. The idea that I wanted you to get today was that there's a whole complex of factors, not just glands, but a whole complex of factors that affect glandular activity and also affect the basic biology and behavior and development of bees.